You're on, Jeff.
Awesome. Thanks so much, Gary, and thanks for the opportunity to come talk about my favorite fish, maybe to study, the thorny skate. If you've been around for the pre-banter, this is a species that I've been studying for over 2 decades, dating back to the undergraduate times, and I really like telling this story because it's a little bit of closure for me. It was a question that I actually investigated for my undergraduate research fellowship and starting in 2002, and it took over 20 years to figure out the answer to the questions that we were asking back then. So it's kind of a personal and professional success story for me. So hopefully I can get you excited about a species that usually doesn't garner a lot of excitement, the thorny skate. So there's a couple projects that I'll talk about that we've done on thorny skate over the past few years, but I'll focus mostly on two recent genetics projects that we've done largely in cooperation with the University of Florida and a ton of geneticists actually from the US and in Europe. So I definitely want to start out by acknowledging all of the people that were involved in this line of research over decades. I've put some logos up on the screen. Sometimes in my excitement, I don't name people by name. I see a lot of people on this screen, these names that have helped me, so thank you. And hopefully you find this cool. All right, so for those of you who are not familiar with thorny skate, they are the lovely skate species list seen here on the left. They are found throughout the North Atlantic. The range is actually quite vast. On the western side, basically down into southern New England, even historically into the mid-Atlantic region, they were found, and the distribution is basically continuous through Greenland, Iceland, the North Sea, the UK, Scandinavia, up into the Arctic Circle. So this is a species that is found over a really vast range, and that's part of the line of questions that we'll get into to try to figure out what does the population structure across this enormous range really look like. So, but looking close to home in the Gulf of Maine, there's a kind of a big problem with thorny skate that's been evident for maybe 20 or even 30 years now, and that is the magnitude and the perpetuation of a really significant population decline. You can see from these two graphics, the biomass in under a couple of different indices has really decreased. I think it's around maybe 95% average decline since the early 1960s. And the one thing that's really interesting is that this decline has persisted with no signs of recovery. And that's not due to lack of effort. So the first attempts to manage skate federally in New England came with the initiation of the Fisheries Management Plan in 2003. And at that time, thorny skate were acknowledged to have been overfished. And as a result, they were listed as a prohibited species. So again, essentially no retention in commercial fisheries was the big action item that came from that FMP. They were also listed as a species of concern in 2004, and since then there's been two petitions to list them under the Endangered Species Act. The second petition, which I'll focus on, was in May 2015, and listing was not found to be warranted because the petition was surrounded around an idea of distinct population segments in thorny skate, and the claim was that their Gulf of Maine was a distinct population segment. And at that time, there were no data available to support that distinction of a DPS in Gulf of Maine. So ultimately, listing was not found to be warranted. The other thing I'll say about this situation is it's kind of peculiar what's going on with thorny skate, because in 2003, both the smooth and barn door skate were also overfished and were listed as prohibited species at that time. However, just in the last year, both smooth and barn door skates were reincorporated into the commercial fishery. So in other words, we've seen a recovery in those species, but for some reason, even though the thorny skate was prohibited at the exact same time, over the last 2 decades, we haven't seen any recovery in thorny skate biomass. And that's kind of been puzzling. And the source of a lot of the research questions that we've pursued is trying to figure out aspects of thorny skate life history, ecology, and population structure that may give clues as to why we haven't seen population recovery. So talking a little bit more about thorny skate in the Gulf of Maine, there's really two main lines of questioning that we followed for the research that I'll talk about today. The first is kind of related to the population structure, the connectivity of thorny skate that are found throughout the Gulf of Maine. So here you see just a plot of where the NAFSC trawl survey caught thorny skate between 2001 and 2011, and you can see that they're pretty widespread throughout the Gulf of Maine, but there's kind of like pockets of activity where there's skates, and then there's areas where they don't exist. And the question within the Gulf of Maine was, what's the connectivity between these groups of skates? Are they freely moving between areas? Are they freely moving between US and Canadian waters? Or are these pockets truly pockets or isolated from one another in any regard? So that's two lines of questioning there to keep in mind. One, what is the connectivity of the skates within the Gulf of Maine? And number two is, are we seeing emigration or immigration of thorny skates basically to the Scotian Shelf and into Canadian waters? The second question of focus for the seminar today is, what is going on with the size morphs of thorny skate that we've known to exist in the Gulf of Maine for probably close to a century. So this picture here shows 3 male thorny skate. On the left, you have a mature male thorny skate. On the right, you have a mature male thorny skate, but in the middle, you have an immature male thorny skate. So this kind of dichotomy in size, or we call it, like we used to say, is 2 different morphs of thorny skate, has always been puzzling because the size discrepancy is huge. And we know that the small morph doesn't eventually turn into the big morph because we see individuals of intermediate size that are immature. So the hypothesis has always been, is this kind of 2 different groups of thorny skate that exist in the Gulf of Maine? Is it a subspecies? Are they different stocks? What is the connection between the large and the small morph, as I'll call them throughout this talk? And there's been a lot of work over the years to try to figure out what the difference is genetically. But until recently, we weren't able to figure it out. So I'll kind of take you through that chronology as well. But first, getting back to that question about connectivity and movements in the Gulf of Maine, geez, about 10 years now, I think we started a satellite tagging study on thorny skate to try to look at short-term movements on the order of up to maybe 10 months to see what their movement patterns were like, or what their seasonal movement patterns may have been, just to try to get an idea of what's going on in the Gulf of Maine, and also to see if we had any emigration from the Gulf of Maine. So for these satellite tags, they're kind of a little bit different from what you typically think, where you get a whole track from an animal from a satellite tag. With these, it was kind of just a point A, point B type thing. So point A is the release location where the skate would be released with the satellite tag. We'd program the tags to stay on the skate for a specific amount of time. The tag would come off. It would give us a reporting location. So basically, this is analogous to a traditional fisheries dependent tag and recapture event where we have point A, point B, you infer linear movements over a time period between two reference points. And that was how the data were structured for this. But the cool thing was because of that, we could kind of pair the satellite tags with a long-standing body of work that did conventional tagging on thorny skate that dates back to when I was an undergraduate doing my undergraduate research at UNH starting in 2002. So in 2016 and 17, we put our over 120 satellite tags on skates. Those are the red dots, the deployment locations that you see on the map on the right. And then working across a lot of individual projects, including the Mass DMF COD industry-based survey, we deployed over 2,000 conventional tags on thorny skate over 17, 18-year period. And all of those deployment locations are in white on the right. So what did we find? Well, thorny skates are maybe lazy or really are homebodies. Essentially, we found that they didn't really move at all. The largest linear displacement over a period of over 9 years was like 48 kilometers. You can see from this map that there's a lot of clustering around the area that they were tagged. There's really no movements between regions. Like, for example, the skates from the west of the Western Gulf of Maine closure didn't really go out to caches. And then the biggest thing is we saw no large scale emigration or migration out of the Gulf of Maine region into Canada. So this is actually consistent with what's been found with regards to thorny skate movement in other places of the range throughout the North Atlantic. On a study in, I believe, the 70s off Newfoundland, the largest distance they observed a skate moving over a decade was 100 kilometers. Same thing within the North Sea. It was pretty localized movements over multiple year recaptures. So it's kind of the same story throughout thorny skates range in that there's not a lot of movement exhibited by this species. And why that's interesting is because it gets back to that idea of the distinct population segment that was kind of the focus of the second Endangered Species Act petition in 2015. And the question is, okay, if we're not seeing a lot of thorny skate movement, even within a region, what is the population structure of these animals throughout their North Atlantic range, which again, as a reminder, basically exists from the Gulf of Maine to Scandinavia, Russia, into the Arctic Circle. So to try to get at the answer to this question, a bunch of us all listed on the right. I won't mention the names specifically, but basically what we wanted to do is collect thorny skate from throughout this whole range and use mitogenome sequencing to look at population structure. So just a reminder, the mitogenome is just mitochondrial DNA, and we're sequencing the entire mitochondrial genome for this portion of the work. So question one was collecting animals from all different regions. You can see here different color circles is all the locations where they sampled. The blocks are the squares or rectangles, whatever. They denote the zones or the regions that we binned them into for analysis. And the question was quite simple, like, what's the relationship genetically between all these regions? The second question that was nested in the mitogenome work was depicted in this photo here of the two size morphs is, can we detect a difference between the morphs using the mitochondrial genome? So first, looking at population structure, mitogenomes were really informative at telling us about broad population structure. The red line, basically separating Newfoundland, Labrador, and Greenland, kind of denotes the demarcation line. So to the east of that line or right of that line, we found that thorny skate exists in a metapopulation that includes places like the North Sea, the Arctic Circle. So basically everything to the right of that line is one group of thorny skate. And then everything to the left or to the west and south is another metapopulation. So we have in the North Atlantic two different genetically distinct groups of thorny skate that we're going to call the Northwest and Northeast metapopulations. The other thing that was interesting was within the Northwest metapopulation, we found there was a lot of similarity genetically between thorny skate and the Gulf of Maine and off Canada, in Labrador and the Grand Bank region specifically. I think it was an order of around 95% similarity. So that was really interesting because right now we're saying, okay, that idea of the distinct population segment kind of holds a little water with the Northwest and the Northeast differentiation, and that the high genetic similarity between the Gulf of Maine and Newfoundland suggests that those kind of are probably the same group of skates and maybe not distinct from one another all that much. But one thing that the mitogenome work could not do was discriminate between the two size morphs. We gave it a valiant effort, but still we weren't finding any evidence of differentiation between the two morphs, and which was a little bit upsetting because we thought it was, we finally had sequenced enough DNA or genetic material to find the difference, but such was not the case. But we weren't discouraged. The group down at University of Florida, led by Dr. Gavin Naylor, just stepped it up and went nuclear. So we figured we couldn't do anything more than sequence the entire thorny skate genome. And if there's some type of genetic basis for those two morphs, it's got to be in there somewhere. So the entire thorny skate genome is over 2.5 billion bases. And now Dr. Pierre Lestrigi, who was then a PhD student in France, went through them, not even as part of his dissertation. It was a side project, which is kind of cool, but also an unbelievably large amount of work. But the work was not in vain. So just to take you through the nuclear whole genome component of this research, we basically did the same thing. We already had sampled thorny skate across all those regions. Again, we had over 500 individuals. We just took 49 of them total and sequenced the entire genome from the same exact regions. And what we were looking for was hidden in a 31 megabase pair, what we're calling a supergene. It's just a section of DNA that has got a ton of genes. You can see it here. It's kind of like this weird anomaly. It has 226 genes in this region of DNA, and there's two notable alleles that are going to hold the key to figuring out the mystery of what's the difference between these two morphs. And I just like to spend a little bit extra time talking about this. Dr. Lestrigi literally went through all the genetic material and just happened to stumble across this bit of DNA on chromosome 2. And he initially thought it was an error, like there was just something that he did wrong. And he was literally about to just discount this section of DNA and move on. And he sent Dr. Gavin Naylor an email, and a couple weeks later, they realized that they definitely should not overlook this section of the DNA because it will solve the mystery that's been plaguing me for over 2 decades. So what exactly is going on in this supergene? Well, there's two different alleles of interest that I'll call HB and HS that are inherited in Mendelian fashion. So you got to think back to your high school or undergrad genetics class, Gregor Mendel, dominance, round versus wrinkled, Punnett square. That's what you need to think about to try to interpret these results. So basically, the small morph of thorny skate has two HS alleles, right? So if you have an HS and an HS together, homozygous recessive, you are a small skate. The large morph of thorny skates have one or two HB alleles. So HB is the dominant allele. So if you're homozygous dominant, HB, you're a big morph of a thorny skate. If you're a heterozygote, an HB and an HS, you're also a large thorny skate. So finally, we have an understanding of what the genetic basis is for these two morphs. So then the next question is, how did this originate and what does it mean for thorny skate? Well, if we kind of look a little bit further, going back to the distribution map of where we sampled the skates, we quickly realized that the HS, the small morph skates, first of all, they're the only morph of skate that exists in the northeast metapopulation. There are no large morph thorny skates that exist basically east of Greenland. So all of this, I don't know if you can see my cursor, but all of these are just small thorny skate. The large morph only exists, therefore, in the northwest metapopulation. And there's a couple of really interesting things here. So these pie charts here represent the kind of allele frequencies or the genotypes of the skates that we sampled in the Gulf of Maine and in Canada. So in Canada, you see there's the pie chart for the red is large, fairly large, and it's much larger than it is in the Gulf of Maine. All right, so what this tells us here is that in the Gulf of Maine, there's a deficiency in heterozygotes, so the HB and HS, and that means that there may be impaired mating that's going on between the large and small morph, because if mating was not impaired, it would be in what we call Hardy-Weinberg equilibrium. Perhaps this is also ringing a bell from your genetics 101 days in your Punnett square. So basically, if you have the ability to interbreed freely, there's going to be an expected number of heterozygotes in the population. And in the Gulf of Maine, we are deficient in heterozygotes. And in Canada, they're not deficient. The population is in Hardy-Weinberg equilibrium. So even though I said before, the US and Canada have a lot of similarity, around 95% genetically based on the mitogenomes, we know that there's different things going on between these regions. The other thing to take away from these pie charts is that you can see they're mostly dominated by like this purple blue color. And that basically means that most of the individuals in these regions are members of the small morph. So in the Gulf of Maine, only around maybe 30% of the animals that we sampled are this large morph. So basically from that, we know that most of the thorny skates that we're seeing in the Gulf of Maine are indeed this small morph, even though the HB allele is dominant. So it's kind of a confusing situation, but it's just sheer numbers thing that there's more small ones. And I'll explain a little bit more about why that's the case in the coming slides. But the question we had was, okay, we kind of know this demographic of allele frequencies and we know where the morphs exist, but where did this HB allele come from? Like, why do we have this weird situation here? And with a little work, we came to the hypothesis that the HB allele came as an introgression from another amblyrogyid skate. We're thinking it's the Arctic skate. So basically what happened is maybe around 150,000 years ago, Arctic skates, I guess, hybridized or mated with thorny skate, the small morph of thorny skate, and some of the Arctic skate DNA became incorporated into the thorny skate genome and gave rise to that large morph. So again, we know that the small morph is the ancestral form of thorny skate, and I guess the large morph is kind of the more derived, and it originated from an introgression of genetic material from the Arctic skate, which is kind of crazy to think about. So I've gone pretty fast through this, and I think we'll have plenty of time for questions, but this last couple slides are just intended to try to bring this all together and try to make sense of it, both evolutionarily as well as in a context of fishery science. Again, thorny skate are not really doing well in the Gulf of Maine. They're not rebuilt. We're trying to figure out why. So hopefully these last two slides will kind of bring everything home and make all this make sense. So based on the mitogenome and the whole genome sequence data, we know that thorny skates originated as a species somewhere around the Greenland, Iceland region. Again, in that side of the Atlantic, it's only the HS allele that's there, and then only the small morph of thorny skate exists in the Eastern Atlantic. That is the ancestral form. Around 900,000 years ago, there was a radiation of the small thorny skates from where they evolved around Greenland and Iceland, both to the southeast to colonize the North Sea, as well as to the west. The movement of the colonization event was 3 times greater to the west than it was to the east. And that is when thorny skates basically colonized what is now the Northwest Atlantic. So Scotian Shelf, Labrador, Grand Banks, and Gulf of Maine. We also now know that after that westward radiation, around 160,000 years ago, is when the exchange of genetic material between the Northwest and the Northeast stopped. And that's when we have now 2 distinct genetic populations of thorny skate that exist throughout the North Atlantic range. Again, we also know that sometime after that separation occurred, that there was the introgression of the genetic material from the Arctic skate, when the Arctic skate hybridized with a small morph, giving rise to the large morph. So how does this all help us with thorny skate population management? Well, there's a bunch of ways. So based on what I told you about the tagging data, we know that there's very little movement going on in the Gulf of Maine. That other study off Canada also showed a limited movement. So we basically shouldn't be expecting recolonization of the Gulf of Maine from thorny skates from other regions. It just doesn't seem to be their MO. They're not a species that migrates a lot. They kind of have just ended up in an area over evolutionary history and existed there, and really are kind of staying there. So where that's kind of an issue is if we have a resident population, right, especially in the Gulf of Maine, we know that the waters of the Gulf of Maine are warming fairly at a fast rate, and there's been a couple interesting studies out of the Northeast Fisheries Science Center that have suggested that thorny skate probably aren't going to do too well with warming ocean conditions. So there's been some contraction of their range within the Gulf of Maine. There's some hypotheses that their distribution will become more fragmented as they redistribute into deeper, colder waters as waters warm. So basically, thorny skate are one of the species that are at highest risk for climate change. And that's kind of a problem when you don't really have the ability or willingness to move away from deteriorating environmental conditions. So maybe that is one reason why we are seeing this, we see this decline and we're not seeing any recovery, because we're not getting any skates coming in from the north. The conditions in the Gulf of Maine are becoming less hospitable for thorny skates, because if you think about their broad distribution, the Gulf of Maine is essentially the southern extent of the Western North Atlantic population range where, impacts are usually first realized with respect to climate change. We also know that in the Gulf of Maine, as I said, we're at a deficiency of heterozygotes between the large and small. So that suggests that there may be impaired mating going on between the morphs. Again, as a reminder, elasmobranchs, including skates, have internal fertilization. So you could make the case that it's physically not possible, or at least physically very challenging for a large and a small thorny skate to mate. So this picture, we have here a large mature male, but the small females, when they're mature, are around the same size, and the clasper length of one of these big mature thorny skates is essentially almost the length of the entire disc of a small thorny skate. So with internal fertilization needing to happen, you kind of have like a physical barrier, a morphological barrier that may just be preventing them from mating. In my mind, I think the only possibility would be if one of these mature small males what mated with a larger female, because that seems to make sense anatomically, but the opposite, a big male with a small female, doesn't seem to be possible. So the take home here is that it's impaired mating may be one of the factors that's contributing to the lack of recovery. If population biomass was driven down in the 60s and 70s from overfishing, now there's maybe so few thorny skate around in the Gulf of Maine that it becomes really challenging for them to find a suitable mate. So not only do you have to find a mature thorny skate, you have to find a mature thorny skate that's of a size that you can mate with. So it just makes it harder, especially at low population biomass, to see recovery. Let's see. All right. So we know from the genetics work that the US and Canadian population of skates are really closely related genetically. But what's really odd is that while population decline of thorny skate has been realized on the Scotian shelf, kind of down in this region, based on Canada DFO survey data, up on the Grand Banks and off Labrador, further to the north and east, where there's a fishery still for thorny skate, the population is actually doing really well. This plot here basically shows that it's in the green. IUCN least concern. It's just kind of a robust population. So the question is, with underfishing pressure, how do we have an unfished population essentially in the Gulf of Maine doing so poorly, and a fished population in the Grand Banks doing so well? And that's definitely a line of additional questioning that we're hoping to look into, but I think some clues can be found in this pie chart here. So I've probably said it a few times, you probably don't remember, but we've got a lot of heterozygotes in this area of Canada, which suggests that it's easier for the large and small morphs to mate up in Canada. And that might be why the population is able to sustain itself in the face of a little bit of fishing pressure, because there actually is less of a barrier between the two morphs and with respect to mating. The one thing that's really interesting, and one line of inquiry that we're hoping to try to follow with some Canadian scientists, is looking at the size overlap between the large and small morphs in like the Labrador, Newfoundland, Grand Banks region. Just looking at some basic morphometrics data that are available, there seems to be more of a overlap between the size range of the small and the large morph, and that greater overlap in a size manner may actually be the thing that allows for mating to be facilitated up there. I didn't mention it, but in the Gulf of Maine, if you think back to the original slide that I showed with the 3 different skates lined up side by side, there's a huge difference in size between the small morph and the large. I'd hypothesize that the small morph doesn't get more than maybe 70 centimeters, and the large morph doesn't even mature until they're 85. So there's a larger gap in terms of size between them, and that may be why we are seeing this inability to mate in the Gulf of Maine, and the ability to have better mating between the morphs in the northern latitudes in Canada. And then lastly, with respect to fisheries management, looking closely into the Gulf of Maine, I think it's pretty logical that continued landings prohibition makes sense. We are 22 years into a 25-year thorny skate rebuilding plan that started in 2003 and is ending in 2028. And we're pretty sure that we're going to fall short of the rebuilding plan. So there's been some discussion starting about what's the recourse? What do we do about that? And continuing the land use prohibition seems like a logical place to start. There's also been some discussion about the benefits of closed areas for thorny skate. If you kind of peer down at this plot here, you can see that a lot of the skates that we tagged in the caches groundfish closure area remained there over periods of up to 10 months to a year. So that homebodiness, the lack of movement, actually may be quite beneficial to thorny skates with respect to existing closed areas. It might be a sanctuary, a haven for them to escape any incidental fishing mortality based on bycatch. And then lastly is just now that we know that we have a pretty large genetic similarity between the US, Gulf of Maine, and Canada, is there reason to try to coordinate with the Canadians and do some kind of joint management, potentially specifically with respect to the Scotian Shelf, which is an area that is now closed. The skate fishery in Canada is now closed there, but again, it's open in the kind of the Grand Banks region. But if we're faced with a third ESA petition for thorny skate, I feel like this might all come to blows and might be really impactful to try to coordinate with the Canadians to look at the preservation of genetic diversity and the whole idea of this distinct population segment when it comes to true extinction risk. So that was a lot. Hopefully you have some questions. Again, thanks to so many people who were involved in this work, putting out over 2,000 conventional tags over 20 years, all the geneticists who worked in the lab, all the people who collected tissue samples from NOAA surveys and research trips, the fishermen who took us out. The list is long and long. I could probably talk another 30 minutes individually about the roles of each one of these people played, but I will not do that. Instead, hopefully I'll talk to you and answer some questions. Thanks a lot.
Thanks, Jeff. That was great. So I'll open it up to questions. If you have a question, either unmute or put something in the chat, and I'll ask the first question. So you mentioned that it's the HB allele, right? That does the morph for the large morph.
Yep.
If that was due to introgression with that Arctic skate, why isn't it still in existence with the northeastern population, but it is in the western population?
All right, let me go back. So the population originated here around Greenland, Iceland, right? We had radiation westward into what we call Northwest Atlantic, right? So this was all small skates and they radiated west to colonize this region. So initially in the Northwest Atlantic, it was only small thorny skate. And then at some point in time, after the small skates had colonized the Northwest Atlantic, that's when the introgression occurred. So it was just kind of like a localized thing that happened after the small morph radiated westward. So because that didn't happen to the east, that's why you see no HB allele in the Northeast. And it also shows, it kind of validates that lack of movement that we've shown with the tagging data, because no HB individuals who only exist over in the Northwest have migrated back east. Does that make sense?
Yeah, so you're thinking that Arctic skate might have come down like through Greenland and down the coast or something.
Yeah, so keep in mind this was rough between 100 and 160,000 years ago. I am not a geologist. I don't know exactly what the continents looked like back then, but it may look a lot different than it does now.
Mhm.
So all we know is that that happened away from the area where the population originated, again, off Iceland and Greenland.
Cool, thanks. All right, questions? Other questions?
Hey, Jeff. Scott here. Hey, you had hypothesized about the maybe a less difference in the morph sizes in Canada versus Gulf of Maine. I wonder also if it has to do with have they looked at growth rates? You know, because if the Canadian fish or if the Canadian skates are growing slower, there may be a they may have more time. The large morphs may have more time as a slightly smaller size, in which the mating may not be impeded quite so badly.
Yes, exactly. That's a great point and a great observation. It seems to be the case. The growth curves in Canada, there's definitely a lot more overlap between. They basically did one giant growth curve and they included all thorny skate in there because there was no way to differentiate them. And you can see that it's a lot of variability, but a lot of overlap. And that is kind of one of the working hypotheses, that there's either more time or that there's just, yeah, exactly what you said, more capabilities of being in the sweet spot because of that. And what we're trying to do now is collect tissue samples from thorny skates and the DFO surveys across their entire latitudinal range from the Scotian Shelf all the way up essentially to the Greenland border to look at size distribution of skates with each genotype. And that hopefully will give us a better idea about what you're talking about. But in the Gulf
Yeah.
of Maine, when we did this aging back in the day, we only did the large ones. So we never estimated a growth curve for the small ones because we never knew what to do with it. And now that's actually one of the things I'm trying to do, because you can you can genotype all the archived samples, discriminate between the morph, and then redo the growth curves.
Yeah, I suspect that the water temperature difference between Gulf of Maine and Canada has something to do with the growth rate differences, which would uh, feed into all of that. So pretty cool.
Yes, sir, you're correct.
Tara.
Thanks, Gary, and thanks, Jeff, for the great talk. Um, I'm wondering, you mentioned that the population is really patchily distributed in the Gulf of Maine. Are the morphs overlapping in space? And what's the difference with the patchiness up in Canada, and how the morphs may overlap up there?
It's a great question. I probably should have mentioned that. So both morphs in the Gulf of Maine are sympatric. They're you find them in the same spot. You'll catch them in the same toe. They basically live side by side. When we did the tagging research, we specifically tagged both morphs, and the movement patterns were the same. In terms of how they're distributed in those patches in the Gulf of Maine, that's another line of inquiry. Now that we can figure out what genotype every skate is, if we take samples from all of the skates, which I'm looking at Dave McElroy's name, we can figure out we can assign them to a genotype and then look at their distribution within the Gulf of Maine, if that makes sense. But historically, it's really hard to do because you kind of need a tissue sample to figure out the morphs, especially for immature individuals. So it'll be really challenging to answer that question using older data. And then your question about Canada, I actually don't really know how the large and small are distributed up there. I would assume it's probably similar based on the fact that they're caught in the DFO trawl surveys. But that's another question that we're trying to ask by sampling the latitudinal gradient in Canada to look at allele frequencies with respect to latitude and then even spatially within the survey region. So great questions.
Thanks.
Micah.
Thanks, Jeff. Learned a lot about thorny skate. It's really interesting. You said that we're like where we are in the world, it's like the southernmost portion of the species range. Do you see the sort of like climate change loser spatial contraction, like the rolling up of the southern end of their range extent in the survey data? And is that sort of the driving part of I get the differences in the allele frequencies, as as you explained, it makes a lot of sense, but even if there was greater intermixing between those distinct subpopulations, is it the population decline largely attributed to us being at the very southern extent of the species?
Yeah, it's a great question. It's possible. And it also makes sense if you look at the North Sea. So on the east side of the Atlantic, the populations that are doing the worst are those at the most southerly latitudes. So that tells us there that there's probably a large environmental effect that we're seeing here. It just may be exacerbated by this anomaly in life history that we're seeing in the Gulf of Maine. So there's got to be an environmental correlate because we're seeing it in the UK as well. And you can see it in the trawl survey data, just the steady decline. But it's trying to figure out the relative contributions. Maybe that's the next question, if it's even possible. I'm not sure it is. But environment definitely has playing a key role. And I think in the trawl survey data, you can see them contracting to the western Gulf of Maine, which is interesting. But based on our satellite tag data, the skates that existed basically west of Stellwagen, west of the western Gulf of Maine closure, experienced the coldest water temperatures of any other skate. So the ones that were tagged in caches towards the Hague line out in the basins, they actually experienced warmer water on average, I'd say. So maybe the skates are contracting west just to get at the last bit of colder water.
Oh, all right. Cool.
Other questions? Oh, Kelly.
Hi, Jeff. I feel like I've already asked a bunch of questions.
You have a question that you haven't asked the last 4 times you've seen this.
I think so, but yeah, thanks for continuing to share such fantastic, comprehensive information on thorny for those of us who have to sit on this skate committee for the council. So my question this time has a little bit different than your presentation, but the diet, is there any, I don't know if there's any recent work on, I guess, what do thorny eat? And then has there been any recent investigations and difference, perhaps relating to differences with the morphs?
So back in our UNH days, there was a graduate student who did diet. I can't remember if she did thornies, maybe Scott would remember, but
I don't.
you don't remember. But I know that smaller thorny skates eat smaller things. Worms like polychaete worms and amphipods and molars. As they get larger, they become more piscivorous. So I'm sure there's data out there, but I don't know specifically. And there probably is in Canada too, but I haven't really looked into the dietary differences between the morphs.
Okay, no worries. I was just curious. Thanks. And thanks again for, yeah, every time I glean some more information, every time I'll hear it another 4 times. That's fine.
Thanks.
I saw Tara's hand up for a second. Did you have another question, Tara?
Yeah, but I should wait until everyone else has had a chance.
No, you were next in line, so.
Okay. Thanks, Gary. Um, with regards to the morphs being more different in the Gulf of Maine, what unique pressures do you think that are down there that could be driving the morphs differentiation? With their patchiness, do you think that the fragmentation could have like a ratcheting effect, like a allopatry that's driving their differentiation? I thought Scott's comment about the growth was pretty interesting too.
Yeah, it's hard to say because we don't really know how long this situation was in existence. Like, was there a greater overlap in size historically, and it just became greater over time? It's really hard to get at that, but I think temperature might play a role, at least in contemporary times, because speaking with the Canadian scientists, they are their working hypothesis is it's that's temperature and i.e. latitude has a lot to do with growth and the overall size of the skates. I don't think that they get as many huge individuals. I think the max size that we see in the Canadian data is in the mid to upper 90 centimeter range total length. And in the Gulf of Maine, I've seen some skates pushing 105, 110 centimeters. So I would guess maybe there's a temperature effect that we're seeing. But again, it's really hard to look at it retrospectively without being able to definitively differentiate between each individual that was measured in a survey, so to speak. So it's a question we can ask more now when we get high resolution spatial information about where these skates exist and then look further into their individual growth rates. Again, I said in Canada, they kind of just lumped them all together, and it's a disastrously looking growth curve because the skates are all genetically different. And in the Gulf of Maine, we basically ignored the small ones because we never knew what to do with them. And back then, I guess we thought that the large ones were going to be the form that was dominant or most prevalent in the Gulf of Maine. And lo and behold, it's actually the opposite. We focused our study on the morph that represents 30% of the individuals as opposed to the morph that represents 70% of the individuals.
Thanks. Interesting.
I had one question in the chat, which I lost, but I believe it was, do thorny skates overlap with other commercially harvested skate species? Right, Eric? I think that's what.
Yes. So if you look back, let's see if I can find yeah, so all you need to do is look at this plot. So Caches was closed because it was a massive fishing spot. West of Stellwagen here is there's a lot of fishing pressure here, tons. There's other skate species that exist in the same area, smooth skates, barn door skates, to a lesser extent, maybe little and winter skates overlap with thornies, but usually the three you get together are thorny, smooth, and barn door. And they definitely exist in areas of intense fishing pressure.
Great. Any more questions before we wrap up? It's noon time. I don't see any. So just wanted to thank you, Jeff. That was great. I'm sure everyone appreciated it too. And I will definitely send you email about Marine Quest, and hopefully you guys can join us.
Sounds good. Thanks everyone for tuning in, and thanks for having me.
Thanks, Jeff.
Have a good day.
You too.